My name is Patrick. I contracted sepsis aged 9 months old, and as a result, I lost all of the fingers on my left hand, the second and third digits on my right hand, and my right leg below the knee. But I survived.
Researchers from UBC have found a new way to determine if a patient has sepsis, even at a very early stage. This could decrease the death rate of sepsis greatly. Today, we have Professor Bob Hancock answering some of our questions about their research.
This is considered to be either a confirmed or a suspected bacterial infection together with immune dysfunction. In other words, the immune system doesn't work properly.
Once inside the body, the bacteria begins to multiply. Then it begins spreading into the bloodstream. Bacteria releases endotoxin, which is a toxin on the cell's membrane. White blood cells react to the presence of the bacteria. White blood cells releases inflammatory substances. The combination of bacteria releasing endotoxins and the white blood cell releasing inflammatory substance is referred as sepsis.
So actually there's no particular bacterium associated with sepsis. Virtually any bacteria can cause sepsis, but it's very rare for a patient to go into multiple organ failure unless they have sepsis. So we consider multiple organ failure to be often a sign or symptom of somebody who's going to acquire sepsis.
Sepsis causes small clots to form in the blood vessels, choking off the tissue. When tissue do not receive enough blood, we release lactic acid, and this causes organs to fail. Here, Professor Hancock explains their hypothesis and how they conducted their study.
I told you a lot of people die from sepsis, up to 30 to 35%, and about half of them die during this hyperinflammatory massive inflammation phase, and about half of them die in this immunosuppressive phase. He suggests that it could be a phenomenon called endotoxin tolerance or cellular reprogramming. We chose only genes that were only expressed in individuals with inflammation as our inflammatory signature, and genes that were only expressed when cells were treated twice with LPS as our endotoxin tolerance or cellular reprogramming signature.
Next, Professor Hancock shows us the equipment they used for their research.
This sequencer is what we call a gigabase sequencer, so it basically, this is actually not the high-end model, but it can sequence equivalent to, I guess, about 20 human genomes at a single time run. We use it for RNA sequencing.
We're working in these, they're called biological safety cabinets, and they prevent the cells from becoming contaminated, since cells can easily become contaminated with bacteria that then destroy the tissue culture cells. So when we're dealing with blood, we would basically work with them under these hoods. We'd use sort of centrifuges to spin off the serum and such like, and so we could just take the blood cells and analyze the blood cells.
Next, Professor Hancock describes what their study means to the future of diagnosing and treating sepsis.
So having a molecular diagnostic would just tell physicians a lot earlier on whether a patient has sepsis, and would allow them to use the most appropriate treatment. So maybe if we can reverse that immunosuppression, then we have a really good chance of a new therapy, a new way of treating sepsis.
Professor Bob Hancock and his team are looking forward to test their findings with a larger number of patients. Hopefully in the near future, this could be used to test sepsis and improve the survival rate of sepsis.